All right. Thank you. I need to stand in the right place. Am I in the right place? And not move, which would be really hard for me. But thank you to the organizers for the invitation and for being part of this. It's really exciting. And I wanted to talk about one project that highlights what the title indicates, which is emerging strategies to reverse prostate cancer therapeutic resistance. And I'll start with saying that I have no conflicts, and also to remind myself that this is really all the work of Yara Rodriguez in the lab, who is a graduate student who hopefully will be finishing soon. And the basic problem is something that maybe some of you are familiar with in prostate cancer, which is that basically with more advanced prostate cancer, the main treatment that we have is androgen deprivation therapy, because prostate cancers are really dependent on androgen. And this is usually can be quite effective, but then, like with many other cancer treatments, you have castration-resistant prostate cancer. And then what has been happening in the field is that we are developing more and more potent inhibitors of this axis, the androgen signaling axis, and they show good effect, but then again, you get resistance. So one of the newer agents is enzalutamide, but there are other drugs that are similar to enzalutamide. So generally, when we say enzalutamide resistance, the subtext of that is that we are also thinking of similar agents that may have similar activity.
I'm very sorry. Let me just try to share again. I can't. Okay. All right.
Hopefully now you're seeing it. Okay, great. So what the goal was to try to use a genome-wide CRISPRa activation strategy to identify genes that when activated will confer enzalutamide resistance. And so what Yara did is use the DCAS9 system. Many of you are familiar, I think more maybe with the knockout, but also probably some of you are familiar that you can actually fuse an activator like BP64 here to the DCAS9 and now bring that into a particular site, specifically with the guide RNA, and that will activate the gene that's right there. And so you get to activate the endogenous gene. And you combine this with also a reporter that would read out androgen receptor signaling. And the main reason for this is that, and I think I have some slides to show you later, that essentially the androgen receptor is so important that even with the enzalutamide resistance, many of the times the tumors actually activate that axis to become resistant. So there is an interest also in understanding more androgen independent mechanisms. So with this reporter, which would read out androgen receptor activity, one could be able to get an idea about whether the particular, I'll skip this and just go to this. The particular gene you identify is androgen receptor pathway relevant. So we have in this screen, I think I'm missing probably a slide from this, which is, and it may show up later, but I want to take a break here and tell you about this. Very important finding clinically from patients. Since the approval of these more potent androgen receptor inhibitors, basically about 1/3 of all the cancers that recur now don't have androgen receptor activity. So they are AR negative, and some of them have neuroendocrine markers. So these are what people call neuroendocrine differentiated prostate cancer, but a good chunk of them actually also don't have neuroendocrine. So people call them so-called double negative. So AR negative, androgen neuroendocrine negative. And this is becoming more and more prominent. And one could make the prediction that as you succeed in making more and more potent inhibitors of the androgen receptor pathway, the tumors will just escape and just, differentiate lineage plasticity and so on, and not depend on androgen receptor anymore. So these AR negative tumors are becoming more important. So in any case, this is the result of the screen that Yara did, where you can see here the guides distribution of the guides at the beginning and with the vehicle treated, and then after the treatment, enzalutamide pressure, you can see there are certain guides that have been enriched here in the red box. And if you look at them, you see that actually AR is one of the top guides. So AR, as I mentioned to you, further activation of AR will allow these cells to do better under enzalutamide. So this kind of validates a little bit the screen. There are also a number of other genes like ID1, Wnt, ID3, Wnt pathway genes that have been well studied to promote enzalutamide resistance that came out of the screen. And you can also see like AR and some of the AR cofactors are more enriched in the GFP positive, which is the AR E reporter that I mentioned to you earlier, and others are the opposite. The guides also with the genome ontology analysis will fall into really pathways that people have zeroed in on as pathways that are important when activated or when disrupted to promote enzalutamide resistance. But what Yara wanted to do was take one gene and focus on it and kind of study it. And understanding these drivers, if you will, or promoters of enzalutamide resistance, obviously we understand the biology, we may understand the pathways and how to target, so we can reverse the enzalutamide resistance, and we can also identify biomarkers that may be able to tell us the tumors that are kind of more susceptible or destined to be resistant to enzalutamide. So PRX2 is a homeobox transcription factor, not really very well studied. It's been implicated in EMT. It's also known to be induced by TGF-beta and has been studied in tumor models in breast and colon, but really nothing in the prostate. So Yara was able to validate this by now coming in and turning on this gene with a guide RNA, again, different guide RNAs. So that's what the SG stand for PRX2, and the NC is normal control or non-targeting control. And you can see that when you turn it on, now the cells are able to grow better in the presence of enzalutamide, the red lines, compared to the control cells. Also in the absence of androgen, which is the CSS, that is charcoal strip serum, so they do better. Again, we grow them as colonies and many other different ways in vitro. Also, if you take cells that have high levels of PRX2, so 22RB1 cells are enzalutamide resistant cells. They have the AR variant, which some of you may know about, that is not targeted by the anti-AR agents, so that gives you intrinsic resistance. If you knock down PRX2 in these cells, then these cells don't do as well, and they are more sensitive to enzalutamide now. You can see the same thing in vivo also. When you grow these, either the PRX2 overexpressing cells or the controls in mice, when tumors are established and then you castrate and then treat with enzalutamide, you can see that the PRX2 overexpression is able to confer resistance. These tumors grow well, and the tumor incidence is also much higher in that group. So one of the first things that you may wonder about is whether this PRX2 is somehow related to the AR pathway, because as I told you, the androgen receptor signaling axis is just so dominant in prostate. So what Yara was able to show is that using AR levels as well as AR scores, I'm showing here data for AR activity scores, the expression of PRX2 is negatively correlated to AR activity score in two different data sets that are shown here. I should check several others. And also in this study where we have RNA-seq and ChIP-seq, you can see that if you look at the cluster of patients based on PRX2 expression, you can see that the ones that have high level of PRX2 generally have less PRX2 binding in the genome, AR binding in the genome, and also have lower AR expression. So this is all suggesting that, PRX2 is negatively correlated to AR, and one of the key questions was whether actually it might be an AR target gene. So looking at patient samples, you get the idea that, yeah, it could be, because if you look at patients that received androgen deprivation, where samples were taken before and after, you can see that generally in a significant manner, the levels of PRX2 in each patient increase after ADT. And we can also see when you look at the PRX2 locus here in link up as also in patient material, you can see AR binding near the PRX2 gene. However, when she tried to kind of validate this and directly show that if you inhibit AR, can you see upregulation of PRX2? She was unable to reproduce that here in link-up cells, so shown here. So there's clearly something missing that the regulation is not straightforward. But to her credit, actually, because this was not my idea, upon reading of the literature, she raised the question of whether, yes, maybe AR is suppressing PRX2, but you do need really an activating signal to really express it, and in the link-up cells, maybe that is missing. And because of the hints from the literature that TGF-beta promotes PRX2 expression, also TGF-beta pathway is very important in enzalutamide resistance. She went and had looked at this. In patient material, you can see both the receptor, TGF-beta receptor, and the ligand are nicely positively correlated with PRX2 in patient material. And in this study here, where you can see that the red in the heat map here is high levels of TGF-beta. You can see the samples that have high levels of TGF-beta and low levels of AR and ones that have high levels of PRX2. So you have like a negative correlation to AR and positive correlation to TGF-beta. And in fact, if you look at cells that do have TGF-beta signaling, these are PC3 cells, they have a SMART3 binding right near that AR binding peak. So then she went ahead and validated this. So as I mentioned to you, link-up cells are not sensitive to TGF-beta because they don't have the receptor. It turns out Jindan has these cells, and she kindly provided them to Yara. These are link-up R2 cells, which have the TGF-beta receptor 2, which is the main signaling receptor. And you can see in those cells, ENZ treatment will turn on PRX2, and TGF-beta will do the same thing. And then you get this additive or synergistic effect with the combination. And you see the same also at the protein level here. So then the model now is that, you have this gene that is positively regulated by the TGF-beta pathway and negatively regulated by AR. So the prediction will be AR negative cells that have active TGF-beta signaling. Those are the cells that will have more PRX2 activity. So the next thing then is, as I told you earlier, that when you put the adenocarcinoma under pressure by inhibiting androgen signaling, some of the tumors will, you get resistance, they will maintain AR or, high AR or mutations. But like I said, about 1/3 will be negative for AR and any other neuroendocrine marker. And then some of them all will have a low AR and negative neuroendocrine. So Yara did look at these groups and found that these so-called DMPC tumors that are negative for both are actually the ones that have the highest levels of PRX2. And in order to study this further, then we looked at the model where we overexpress PRX2 and then did RNA-seq with enzalutamide or without, and looked at the different gene expression profiling. And here selected just under baseline genes that are mostly induced, the most significantly induced by PRX2. She got a list of 21 genes that then formed a signature that was then used to stratify patients. So these genes will cluster with each other separately from neuroendocrine signature genes and separately from AR signature genes. And if you look at here, stand up to cancer, which is a metastatic castration-resistant prostate cancer patient cohort, these genes kind of identify a separate group of patients that are distinct from more AR high or neuroendocrine high. So these are the double negative patients. Interestingly, actually, if you take the double negative patients and you look at the score based on PRX2 target gene expressions, the ones that have higher activity of PRX2 do worse. So then if you went ahead to kind of validate this in cell models, so D145 cells are used as a model for double negative prostate cancer. When you knock down PRX2, you can see you decrease colony formation in this. But I think one of the nicer models that she used is this model, which is a double negative prostate cancer patient-derived xenograft type model that's derived from the lab C9 model. So AD is androgen dependent, AI is androgen independent. So lab C9 AI, she was able to show that, in fact, when this PDX goes to androgen independence and enzalutamide resistance, you have an increase in PRX2. You can see decrease in AR target genes like TLK3, and really not much also in synaptophysin, which is neuroendocrine marker. So you can grow these as organoids in vitro. And when you knock down PRX2, you see you decrease organoid size, and also in vivo, with the knockdown, you can see these tumors grow much slower. So what does PRX2 really do? What pathways does it engage, especially in the context of enzalutamide? Okay, so those may be what are important for enzalutamide resistance. So as I told you, we did this RNA-seq, and here NC is the non-targeting control link-up cells, D is DMSO, and E is enzalutamide. So you can see that it's a cluster of genes that are suppressed by enzalutamide, and then another cluster that is induced by enzalutamide, okay? So PRX2, basically, when you overexpress PRX2, you can see that even with enzalutamide, you are not shutting down this particular cluster, because actually PRX2 is activating this gene. So you still have quite a bit of expression of these genes, which are more down-regulated in the control. And down here, you can see the induction in the PRX2 with enzalutamide is higher with these particular genes. Okay, and these are all generally all upregulated by PRX2, regardless of ENZA. And very few genes are actually repressed by PRX2. And the one that came out is the CDKN2A, which is a cell cycle inhibitor. So as you might expect, or it makes sense that a lot of these genes that PRX2 is upregulating are genes that are important for cell cycle progression. And down here, some of these genes that are induced by ENZA, actually some of the bad genes that ENZA will induce, which are actually pro-tumorigenic. Like many people have studied these, and many of these would promote ENZA resistance. So ENZA is inducing its own kind of weakness, if you will. And these are amplified or increased by PRX2. So overall, it makes a lot of sense that with high PRX2, ENZA is less effective. IR also did some ATAC-seq and integrated with RNA-seq. I look at the pathways, and we focused on two pathways that came out. So in addition to the TGF-beta, for this is E2F, transcription factor network, and BCL2. And basically here, just validating that bottom line is when you have high levels of PRX2, it's kind of mimicking in activation with RB, basically. Okay, but you can see the phosphorylation RB is changed and so on. That's what she shows here. And consistently also the CDK4/6 inhibitor is actually more effective in the PRX2 overexpressing cells compared to controls. So that's one pathway. And then also BCL2 is more upregulated in the presence of activation of PRX2, especially in the context of ENZA or castration, charcoal strip serum. And these cells will respond to BCL2 inhibitor, and in combination with ENZA, you have a better effect. So and you can see this also with organoids. So we have these two pathways that are all dysregulated by PRX2 that are potentially targeted. One is the RB-E2F CDK4 pathway, and the other is the BCL2. So Yara did in vitro studies first, and then did this really big in vivo study to test this idea. So these are link-up SG PRX2 cells, and tumors are established. And when they are established, they will castrate. When they grow back, now you start the treatment, okay? With vehicle, you can see that's the red line here, they grow. With ENZA, ENZA doesn't touch these tumors. So these are really fully ENZA resistant according by this criteria. When you add the BCL2 inhibitor, or the CDK4/6 inhibitor, which are these two lines here, you do suppress tumor growth, and the combination, you have a stronger effect. So the idea is that the combination of CDK4/6 inhibitor like palbociclib and BCL2 pathway inhibitor like venetoclax should be able to reverse enzalutamide resistance in this kind of patients that have high level of PRX2. And if you look at the tumors, as you might expect, you have decreased proliferation significantly with the CDK4/6 inhibitor and increase in apoptosis in the tumors that got the BCL2 inhibitor. So you got both effects in the combination, and this could be part of what is mediating the response. So the summary of this is that just in an unbiased way, using the CRISPR activation genome-wide screen, we're able to find genes that, when overexpressed, will make these cells more resistant to enzalutamide. And we have studied one of them in depth, this PRX2, shown that how it is dysregulated in human tumors, especially in the double negative prostate cancer, and that this gene actually is under regulation of TGF-beta and AR, and also will activate pathways that feed into the cell cycle E2F and survival BCL2, and this could be targeted to reverse enzalutamide resistance. So as I mentioned, we know this is all a work led really by Yaro Rodriguez, and with really a lot of help from all the members of the lab, as shown here, and a lot of help also from Jindan, Debu, and Daniela. And also the idea for this actually was part of our PCF grant with MAHA, and it kind of took off and developed a life of its own. So I also acknowledge her and the funding sources. Thank you.
Great talk, Sarki, as always.
Thank you.
So I noticed your on the seq data showed the your PRX2 overexpression cells reversed the gene expression that was altered by ENZ. I don't even you notice that. So I'm asking. My question is: so androgen inhibits PRX2, and PRX2 is upregulated in ENZ-resident cells. The question is: does PRX2 actually inhibit? It actually activates AR signaling?
So so that model, remember, is where you actually are using the guide RNA to turn on the PRX2. So the way we are thinking of it is you are taking it away from the regular endogenous regulation. So it's like when you try exogenous overexpression, basically, right? So the circuit regarding how AR will be repressing that and all that is not there in that setting, okay? So, but I think what you are getting at is that whether we looked at the other way, whether PRX2 is feeding back, right, to regulate AR.
Yeah, I thought that your on-seq data and also your linker plus PRX2 data was totally responsive to ENZ. I think that data was amazing. So I was wondering, does that actually activate AR?
Yeah, so I don't remember. Yara, you are here. Do you remember if we looked into that? No? No, we do. Yeah. So that's something we haven't really specifically looked at that, but that's a good point.
The recovery of the genes were amazing. So yeah. I think it's worth it to look at.
Yes. Thank you.
Just a reminder to please repeat the questions. Thank you.
Oh, sorry. Yeah. Okay. I think the question Jindan was asking was whether PRX2 was regulating AR, because based on the RNA-seq data, it suggests that that may be happening. Yeah.
We said that so the comparison between the gene PRX2 overexpression in basal level, we did see that some of the androgen pathway genes were downregulated in the PRX2 expression cells.
Okay, so to repeat that, looking at the PRX2 overexpression cells and the controls, if you look specifically at some of the AR target genes, they do go down. Yeah.
So in the putting it in the perspective of the translational bridge symposium, are you planning to do any clinical trial with this?
Yes, so we with David Vanderweel, we have been discussing this. Actually, we have been talking to CTEP at the NCI, and they have told us some things that we are working on that. Yes, we plan to do that. Yeah, it would be nice.
It would be a wonderful opportunity.
Yes.
Thank you. Yeah, great effects you see. I think that there are multiple mechanisms for enzalutamide resistance, right? So is there a way, following up on Daniela's question, to identify the patient who will most likely benefit from this combination therapy?
Yeah. So our suggestion will be, first of all, double negative prostate cancer patients, because that's really the group that we have really studied more. And then we have this PRX2 signature. So our suggestion would be like you look at the PRX2 signature, the ones that have high PRX2 signature then should respond to the pathway. So the question is,
What percent of the patients you think have the high signature?
I should know that. Is it 25%? Yeah. Yeah. So what percentage of patients have that signature was the question. Yeah.
We do not.
Okay.